Molecular analysis using UV. ChatGPT said. Molecular analysis using UV spectroscopy. Ultraviolet UV spectroscopy is a fundamental technique in molecular biology used to analyze nucleic acids, proteins, and other biomolecules. It is based on the absorption of ultraviolet light by molecules containing conjugated double bonds or aromatic rings. The primary application of UV spectroscopy in molecular biology is the quantification and purity assessment of nucleic acids and proteins. Principle of UV spectroscopy. UV spectroscopy relies on the Beer-Lambert law, which states that absorbance is directly proportional to the concentration of the absorbing substance. Molecules absorb UV light at specific wavelengths, depending on their electronic structure. The two major biomolecules analyzed using UV spectroscopy are nucleic acids DNA, RNA, and proteins. UV spectroscopy for nucleic acid analysis 1. Quantification of DNA and RNA. Nucleic acids absorb UV light due to their purine, adenine, guanine, and pyrimidine, cytosine, thymine, uracil bases. The maximum absorbance lambda max for DNA and RNA is at 260 nanometers, A260. The concentration of nucleic acids is determined using the formula: DNA concentration microgram per milliliter equals A260 times 50. RNA concentration microgram per milliliter equals A260 times 40. 2. Purity assessment of DNA and RNA. The ratio of absorbance at 260 nanometers to 280 nanometers, A260, A280, is used to determine purity. A260, A280 to 1.8 indicates pure DNA. A260, A280 approximately 2.0 indicates pure RNA. A lower ratio suggests protein contamination due to protein absorbance at 280 nanometers. The A260, A230 ratio is also checked for contaminants like phenol, carbohydrates, or salts. A ratio in GT, 2.0 indicates high purity. UV spectroscopy for protein analysis 1. Protein quantification. Proteins absorb UV light due to the presence of aromatic amino acids tryptophan, tyrosine, and phenylalanine. The maximum absorbance for proteins occurs at 280 nanometers, A280. Protein concentration is calculated using A280 and an extinction coefficient specific to the protein. Alternatively, the Bradford, Lowry, and BCA assays use UV visible absorbance at different wavelengths for protein estimation. 2. Protein purity and contamination. The A260, A280 ratio helps identify nucleic acid contamination in protein samples. A260, A280 and GT, 1.5 suggests nucleic acid contamination in a protein sample. Applications of UV spectroscopy in molecular biology. Nucleic acid quantification, used before PCR, sequencing, and cloning experiments. Protein quantification, essential for enzyme assays, electrophoresis, and Western blotting. Purity assessment, ensures high-quality DNA, RNA for molecular biology applications. Detection of structural changes, UV spectra can reveal protein denaturation or DNA-protein interactions. Previously asked CSIR net question. CSIR NET 2020. Which wavelength is used for nucleic acid quantification using UV spectroscopy? A. 280 nanometers. B. 230 nanometers. C. 260 nanometers. D. 320 nanometers. Answer: C. 260 nanometers. CSIR NET 2018. If a DNA sample shows an A260, A280 ratio of 1.3, what does it indicate? A. Pure DNA sample. B. RNA contamination. C. Protein contamination. D. Salt contamination. Answer: C. Protein contamination. CSIR NET 2016. Which aromatic amino acid contributes most to protein absorbance at 280 nanometers? A. Tyrosine. B. Tryptophan. C. Phenylalanine. D. Histidine. Answer: B. Tryptophan. UV spectroscopy remains a vital tool for molecular analysis, providing a rapid, non-destructive method for quantifying and assessing the purity of biomolecules. You said, visible, ChatGPT said. Molecular analysis using visible spectroscopy. Visible spectroscopy, also known as colorimetric analysis, is widely used in molecular biology to quantify biomolecules, detect enzymatic reactions, and study biomolecular interactions. It is based on the absorption of visible light 400-700 nanometers by colored compounds or chromogenic reactions. Principle of visible spectroscopy. Like UV spectroscopy, visible spectroscopy follows the Beer-Lambert law, where absorbance is proportional to the concentration of the analyte. Some biomolecules naturally absorb visible light, while others require chromogenic or fluorogenic dyes for detection. Applications of visible spectroscopy in molecular biology 1. Protein quantification. Since proteins do not strongly absorb visible light, colorimetric assays are used. Bradford assay 595 nanometers uses Coomassie brilliant blue dye, which binds to proteins and shifts absorbance from 465 nanometers to 595 nanometers. Lowry assay 750 nanometers measures protein concentration based on copper ion reduction in a full and Choquel-2 reagent reaction. Bisinchonic acid BCA assay 562 nanometers uses bisinchonic acid to detect copper ion reduction in proteins. 2. Nucleic acid quantification. DNA and RNA do not absorb visible light, but colorimetric dyes are used for detection. Diphenylamine assay detects DNA by forming a blue complex 595 nanometers. Orsinol assay detects RNA by forming a green complex 660 nanometers. 3. Enzyme activity assays. Enzymatic reactions producing colored products are monitored using visible spectroscopy. Alkaline phosphatase AP assay uses p-nitrophenyl phosphate NEP, which turns yellow at 405 nanometers. Beta-galactosidase latch assay uses O-nitrophenyl beta-D-galactopiranocide ONPG, forming a yellow product at 420 nanometers. Peroxidase-based assays use TMB3, 3 feet, 5 feet tetramethylbenzidine to generate a blue color at 450 nanometers. 4. Detection of metabolites. Many metabolic assays rely on chromogenic reactions for detection. Glucose estimation uses the glucose oxidase peroxidase GODPOD assay, forming a pink color at 540 nanometers. Lactate dehydrogenase LDH assay measures NADH reduction, monitored at 490 nanometers. 5. Colorimetric detection in ELISA. Enzyme-linked immunosorbent assay ELISA uses enzyme-substrate reactions to produce colored products, typically measured at 450 nanometers TMV substrate or 405 nanometers NEP substrate. Previously asked CSIR net question CSIR NET 2021. Which protein estimation method uses Coomassie Brilliant Blue dye? A. Lowry assay. B. BCA assay. C. Bradford assay. D. ELISA. Answer: C. Bradford assay. CSIR NET 2019. Which enzyme is used in the colorimetric glucose estimation assay? A. Amylase. B. Glucose oxidase. C. Lactase. D. Peroxidase. Answer: B. Glucose oxidase. CSIR NET 2017. Which wavelength is commonly used to detect ELISA reactions using TMV substrate? A. 405 nanometers. B. 450 nanometers. C. 595 nanometers. D. 750 nanometers. Answer: B. 450 nanometers. Visible spectroscopy is a crucial tool in molecular biology for detecting and quantifying biomolecules, enzyme activities, and molecular interactions through colorimetric methods. You said, fluorescence, ChatGPT said. Molecular analysis using fluorescence spectroscopy. Fluorescence spectroscopy is a powerful technique used in molecular biology to study biomolecules, detect specific proteins and nucleic acids, and analyze molecular interactions. It relies on the absorption of light at a specific wavelength and the subsequent emission of light at a longer wavelength. Principle of fluorescence spectroscopy. A fluorophore absorbs light at an excitation wavelength and emits light at a longer emission wavelength due to energy loss in the excited state. The difference between excitation and emission wavelengths is known as the Stokes shift. The intensity of fluorescence is proportional to the concentration of the fluorophore, allowing for sensitive detection. Applications of fluorescence spectroscopy in molecular biology. 1. Nucleic acid quantification. Fluorescent dyes enhance the sensitivity of DNA, RNA detection compared to UV spectroscopy. SYBR green and ethidium bromide ETBR intercalate between DNA bases and fluoresce upon UV excitation. Hoaxed and DAPI dyes bind to AT-rich regions of DNA and are used in cell imaging. 2. Protein quantification and analysis. Fluorescent protein labels, green fluorescent protein GFP, and variants like mCherry and YFP allow real-time protein tracking. Fluorescent dyes, FITC, fluorescein, TRITC, rhodamine, and alexafluor dyes conjugated to antibodies enable detection in Western blotting and immunofluorescence. Tryptophan fluorescence, intrinsic fluorescence of tryptophan residues in proteins can be used to study protein folding and conformational changes. 3. Fluorescence microscopy and imaging. Immunofluorescence microscopy uses fluorophore conjugated antibodies to visualize cellular components. Fluorescence in situ hybridization FISH detects specific DNA, RNA sequences and chromosomes using fluorescently labeled probes. Live cell imaging, fluorescent proteins and dyes enable real-time observation of cellular processes. 4. Flow cytometry. Cells labeled with fluorescent markers are analyzed based on light scattering and fluorescence intensity. Used for cell sorting, FACS, immune cell phenotyping, and apoptosis detection. 5. Fluorescence resonance energy transfer FRET measures interactions between biomolecules based on energy transfer between two fluorophores. Used in protein-protein interaction studies, biosensors, and molecular diagnostics. 6. Quantitative PCR, qPCR, and ELISA. Real-time PCR, qPCR, uses SYBR green or Techman probes to quantify nucleic acids. Fluorescence-based ELISA, more sensitive than colorimetric ELISA, detecting proteins with higher specificity. Previously asked CSIR net question CSIR NET 2021. Which dye is commonly used for real-time PCR fluorescence detection? A. Ethidium bromide. B. SYBR green. C. DAPI. D. Rhodamine. Answer: B. SYBR green. CSIR NET 2019. Which fluorophore is used for live cell imaging? A. Hematoxylin. B. GFP. C. Crystal violet. D. Safranin. Answer: B. GFP. CSIR NET 2017. Which principle is used in FRET-based protein interaction studies? A. Energy transfer between two fluorophores. B. Light scattering by proteins. C. Absorbance at 280 nanometers. D. Nucleic acid hybridization. Answer: A. Energy transfer between two fluorophores. Fluorescence spectroscopy provides high sensitivity and specificity for molecular analysis, making it an essential tool in modern biological research. You said. Immunofluorescence microscopy. ChatGPT said. Immunofluorescence microscopy. Immunofluorescence microscopy is a powerful technique used to visualize specific proteins or cellular structures using fluorescently labeled antibodies. It is widely used in cell biology, pathology, and molecular diagnostics for detecting proteins in cells and tissues. Principle of immunofluorescence microscopy: a fluorophore conjugated antibody binds to a specific antigen in a sample. The sample is illuminated with light of a specific wavelength, exciting the fluorophore. The fluorophore emits light at a longer wavelength, which is detected under a fluorescence microscope. Different fluorophores FITC, TRITC, alexafluor, DAPI allow multicolor labeling of multiple targets in a single sample. Types of immunofluorescence microscopy. 1. Direct immunofluorescence DIF. A primary antibody is directly labeled with a fluorophore and binds to the target antigen. Advantages: faster, fewer steps, less background noise. Limitation: lower signal intensity due to fewer fluorophores per antigen. 2. Indirect immunofluorescence IIF. A primary antibody binds to the target antigen, and a secondary antibody conjugated to fluorophore binds to the primary antibody. Advantages: higher sensitivity due to multiple secondary antibodies binding to each primary antibody, amplifying the signal. Limitation: requires more steps, increasing the risk of background noise. Applications of immunofluorescence microscopy. 1. Cellular and subcellular protein localization detects specific proteins in cells or tissues, helping study cellular organization. Example: localization of tubulin, cytoskeleton, actin filaments, or nuclear proteins. 2. Disease diagnosis and pathology. Used in autoimmune disease detection, e.g., antinuclear antibodies ANA in lupus. Detection of viral infections like herpes simplex virus (HSV) in tissue samples. 3. Cancer research and biomarker detection identifies cancer-specific proteins in tissues, aiding in diagnosis and prognosis. Example: detection of HER2 in breast cancer or P53 in tumors. 4. Microbial and viral detection. Used to visualize bacterial, fungal, and viral antigens in clinical samples. Example, detection of Mycobacterium tuberculosis or HIV proteins in infected cells. 5. Neuroscience research. Visualizes neurotransmitter receptors, synaptic proteins, and neuronal structures. Example, staining for GFAP, glial fibrillary acidic protein, to study astrocytes in the brain. Limitations of immunofluorescence microscopy photobleaching. Fluorophores degrade over time with prolonged light exposure. Non-specific binding. Background fluorescence can interfere with interpretation. Requires specialized equipment. High-resolution fluorescence microscopes are expensive. Previously asked CSIR Net question CSIR NET 2021. Which of the following is a major advantage of indirect immunofluorescence over direct immunofluorescence? A. Reduced background fluorescence. B. Higher sensitivity due to signal amplification. C. Faster detection time. D. No need for secondary antibodies. Answer. B. Higher sensitivity due to signal amplification. CSIR NET 2019. Which fluorophore is commonly used for nuclear staining in immunofluorescence microscopy? A. FIT. B. Rhodamine. C. DAPI. D. Alexafluor 488. Answer. C. DAPI. CSIR NET 2017. Immunofluorescence microscopy is commonly used to detect which of the following? A. DNA methylation patterns. B. Membrane potential of mitochondria. C. Localization of proteins within cells. D. RNA polymerase activity. Answer. C. Localization of proteins within cells. Immunofluorescence microscopy is a highly specific and sensitive method for visualizing molecular components in biological samples, making it invaluable for research and diagnostics. You said. Detection of molecules in living cells. ChatGPT said. Detection of molecules in living cells. The detection of biomolecules in living cells is crucial for understanding cellular functions, signaling pathways, and disease mechanisms. Various techniques allow real-time monitoring of proteins, nucleic acids, metabolites, and ions within living cells without disrupting their physiological state. Key techniques for detection of molecules in living cells. 1. Fluorescence microscopy and live cell imaging. Uses fluorescent dyes, genetically encoded fluorescent proteins, or labeled antibodies to detect specific biomolecules in live cells. Example, green fluorescent protein GFP tagging allows visualization of proteins in real time. 2. Fluorescence resonance energy transfer FRET. Measures interactions between biomolecules using two fluorophores that transfer energy when they are close together. Used to study protein-protein interactions, enzyme activity, and receptor signaling. Example, FRET-based calcium sensors detect calcium ion concentration changes in neurons. 3. Bioluminescence imaging BLI. Uses bioluminescent proteins like luciferase to detect molecules in live cells. Highly sensitive for tracking gene expression and cellular metabolism. Example, Firefly luciferase reporters in cancer research to track tumor growth in live cells. 4. Fluorescent biosensors. Genetically engineered molecules that change fluorescence in response to a specific cellular condition, e.g., pH, ATP, Ca² levels. Example, genetically encoded calcium indicators GECIs, like GCAMP detect calcium flux in neurons. 5. Single molecule imaging SMI. Uses super-resolution microscopy to visualize individual molecules in living cells. Example, tracking individual mRNA molecules using MS2 GFP tagging. 6. Fluorescence-activated cell sorting FACS uses flow cytometry to detect and sort live cells based on fluorescent signals. Detect specific molecules on or inside cells using fluorophore-labeled antibodies. Example, sorting immune cells based on CD markers. 7. Raman spectroscopy and surface-enhanced Raman spectroscopy SIRS label-free method to detect molecular fingerprints inside live cells. Detects proteins, lipids, and nucleic acids without requiring fluorescence. Example, monitoring drug accumulation in cancer cells. Applications of live cell molecular detection. Cellular signaling studies, detecting kinase activity, calcium waves, and neurotransmitter release. Cancer research, tracking oncogene expression, apoptosis, and drug response. Neuroscience, monitoring synaptic activity, ion flux, and neurodegenerative disease markers. Stem cell research, identifying pluripotency markers and differentiation pathways. Pathogen detection, identifying viral proteins, bacterial toxins, and immune responses in infected cells. Previously asked CSIR Net question CSIR NET 2022. Which technique allows real-time tracking of protein movement in live cells? A. Western blotting. B. Fluorescence recovery after photobleaching FRAP. C. ELISA. D. Immunohistochemistry. Answer, B. Fluorescence recovery after photobleaching FRAP. CSIR NET 2020. Which fluorescent protein is commonly used to study protein localization in live cells? A. Luciferase. B. GFP. C. DAPI. D. Ethidium bromide. Answer, B. GFP. CSIR NET 2018. Which technique is best suited to detect intracellular calcium levels in live cells? A. Mass spectrometry. B. FRET-based biosensors. C. SDSPAGE. D. Southern blotting. Answer, B. FRET-based biosensors. The ability to detect molecules inside living cells is essential for understanding dynamic cellular processes and developing new therapeutic strategies. You said. Circular dichroism. ChatGPT said. Circular dichroism CD spectroscopy. Circular dichroism CD spectroscopy is a biophysical technique used to study the secondary structure, folding, and conformational changes of biomolecules, primarily proteins, nucleic acids, and chiral molecules. It is widely applied in structural biology, protein chemistry, and pharmaceutical research. Principle of circular dichroism. CD spectroscopy measures the difference in absorption of left and right circularly polarized light by a chiral molecule. Chiral molecules interact differently with circularly polarized light, producing a CD spectrum. The shape and intensity of the CD spectrum provide information about the structural features of biomolecules. Applications of circular dichroism spectroscopy. 1. Protein secondary structure analysis. CD spectra can determine the proportion of alpha helices, beta sheets, and random coils in proteins. Example, alpha helix-rich proteins show strong negative bands at 208 nanometers and 222 nanometers, while beta sheets show a negative band at 218 nanometers. 2. Protein folding and stability studies. CD is used to monitor protein unfolding and refolding under different conditions like temperature, pH, or denaturants. Helps in studying the effects of mutations or ligand binding on protein conformation. 3. Nucleic acid structure analysis. CD spec to reveal A form, B form, or Z form DNA and detect RNA folding patterns. Example, bDNA exhibits a positive peak at 275 nanometers and a negative peak at 245 nanometers. 4. Ligand binding studies. CD can detect conformational changes upon drug or ligand binding to proteins or nucleic acids. Example, CD is used to study protein-ligand interactions in drug design. 5. Structural comparison of proteins. Used to compare the conformation of recombinant proteins with their native counterparts. Essential for biopharmaceutical quality control, e.g., biosimilar drugs. 6. Monitoring protein aggregation. Aggregation-related changes in secondary structure can be tracked using CD. Useful in studying neurodegenerative diseases like Alzheimer's and Parkinson's. Advantages of CD spectroscopy: non-destructive, does not alter the sample. Rapid and sensitive, provides structural information in minutes. Requires small sample volumes, ideal for studying biomolecules in solution. Can be used under physiological conditions, studies proteins in their native environment. Limitations of CD spectroscopy. Requires optically pure samples, contaminants can interfere with measurements. Limited resolution, does not provide atomic level details like X-ray crystallography or NMR. Signal intensity is weak, requires a high concentration of biomolecules for accurate detection. Previously asked CSIR net question CSIR NET 2021. Which type of biomolecular structure can be analyzed using circular dichroism, CD spectroscopy? A. Primary structure of proteins. B. Secondary structure of proteins. C. Tertiary structure of proteins. D. Quaternary structure of proteins. Answer: B. Secondary structure of proteins. CSIR NET 2019. Which of the following is a characteristic CD signal for alpha helical proteins? A. Positive peak at 275 nanometers. B. Negative peaks at 208 nanometers and 222 nanometers. C. Positive peak at 218 nanometers. D. No CD signal in the far UV region. Answer: B. Negative peaks at 208 nanometers and 222 nanometers. CSIR NET 2017. Which structural transition can be monitored using CD spectroscopy? A. Alpha helix to beta sheet conversion in proteins. B. DNA replication. C. Protein phosphorylation. D. Protein synthesis. Answer: A. Alpha helix to beta sheet conversion in proteins. Circular dichroism spectroscopy is a valuable tool for studying biomolecular structure, folding, and interactions, making it essential in both fundamental and applied research. You said, fluorescence, ChatGPT said. Fluorescence and its applications in molecular biology. Fluorescence is a phenomenon where certain molecules, called fluorophores, absorb light at a specific wavelength and then emit light at a longer wavelength. This property is widely used in biological and biochemical research for detection, imaging, and quantification of biomolecules. Principle of fluorescence. A fluorophore absorbs light excitation at a specific wavelength. The excited molecule reaches a higher energy state. The molecule returns to its ground state, emitting light fluorescence at a longer wavelength than the absorbed light. The difference between the excitation and emission wavelengths is called the Stokes shift, which helps in detecting fluorescence signals. Fluorescence-based techniques. 1. Fluorescence microscopy. Uses fluorophore-labeled molecules to visualize specific structures inside cells. Applications: protein localization, cellular dynamics, and live cell imaging. Example: GFP-tagged proteins allow real-time observation of protein movement in cells. 2. Immunofluorescence (IF). Uses fluorophore-conjugated antibodies to detect specific proteins in fixed cells or tissues. Types: directive, primary antibody is labeled with a fluorophore. Indirective, uses an unlabeled primary antibody and a fluorophore-labeled secondary antibody. Application: diagnosing autoimmune diseases by detecting antibodies in patient samples. 3. Fluorescence-activated cell sorting (FACS), a flow cytometry technique that uses fluorescence to analyze and sort cells. Applications: identifying immune cells, detecting cancer markers, and isolating stem cells. 4. Fluorescence in situ hybridization. FISH uses fluorescent probes to detect specific DNA or RNA sequences in cells. Application: identifying chromosomal abnormalities in genetic disorders and cancer diagnostics. 5. Forster resonance energy transfer (FRET) measures molecular interactions by detecting energy transfer between two fluorophores. Application: studying protein-protein interactions and enzyme activity. 6. Fluorescence recovery after photobleaching (FRAP) analyzes protein mobility by bleaching a fluorescent signal in a region and tracking recovery. Application: studying membrane protein dynamics. 7. Fluorescence polarization (FP) measures molecular interactions based on changes in fluorescence polarization. Application: drug discovery and receptor ligand binding studies. 8. Single molecule fluorescence detects individual biomolecules, providing high-resolution insights into molecular behavior. Application: studying transcription factor binding to DNA. Applications of fluorescence in biological research: cell imaging, detecting organelles, proteins, and nucleic acids. Molecular diagnostics, identifying infectious diseases, e.g., tuberculosis using fluorescent probes. Cancer research, studying tumor markers and metastasis. Neuroscience, monitoring neurotransmitter release using calcium-sensitive fluorescent dyes. Genetics, identifying mutations and chromosomal abnormalities. Previously asked CSIR net question CSIR NET 2021. Which of the following techniques is based on fluorescence and is used to detect protein-protein interactions? A. Western blot. B. ELISA. C. FRET. D. PCR. Answer: C. FRET. CSIR NET 2019. Which fluorophore is commonly used for live cell imaging? A. Ethidium bromide. B. DAPI. C. GFP. D. Kumasi blue. Answer: C. GFP. CSIR NET 2017. Which technique uses fluorescently labeled probes to detect specific DNA sequences? A. Northern blot. B. Western blot. C. FISH. D. EMSA. Answer: C. FISH. Fluorescence-based techniques are essential tools in modern molecular biology, allowing researchers to visualize and quantify biomolecules with high sensitivity and specificity. You said, NMR. ChatGPT said. Nuclear magnetic resonance (NMR) spectroscopy. Nuclear magnetic resonance (NMR) spectroscopy is a powerful biophysical technique used to study the structure, dynamics, and interactions of biomolecules like proteins, nucleic acids, and small metabolites. It provides atomic-level resolution and is widely used in structural biology, drug discovery, and metabolomics. Principle of NMR spectroscopy. Certain atomic nuclei, e.g., H cubed C to the power of 5n cubed V, possess nuclear spin, which aligns in an external magnetic field. When exposed to radio frequency RF pulses, these nuclei absorb energy and transition between spin states. The relaxation of nuclei back to their original state releases energy, which is detected and analyzed to generate an NMR spectrum. The chemical environment of each nucleus influences its chemical shift delta, which provides structural information. Types of NMR spectroscopy. 1. HNMR, proton NMR. Used for analyzing organic molecules, proteins, and metabolites. Provides information on chemical structure, hydrogen bonding, and functional groups. Chemical shifts range from 0 to 10 ppm for organic compounds. 2. Cube C NMR, carbon-13 NMR, detects carbon atoms in biomolecules. Helps in determining molecular frameworks and distinguishing between different carbon environments. Typically uses proton decoupling to simplify spectra. 3. To the power of 5n and cube P NMR. To the power of 5n NMR, used in protein structure analysis and nitrogen-containing biomolecules. Cube P NMR, essential for studying nucleotides, ATP, phospholipids, and phosphorylation processes. 4. 2D NMR, two-dimensional NMR. Provides detailed structural information by correlating signals from different nuclei. Common techniques. COSY correlation spectroscopy identifies spin-spin coupling between protons. HSQC heteronuclear single quantum coherence correlates H with cube C or to the power of 5n. NOESY nuclear Overhauser effect spectroscopy provides distance constraints for protein structure determination. 5. Solid-state NMR. Used for studying membrane proteins, fibrils, and insoluble biomolecules. Requires specialized sample preparation and techniques like magic angle spinning (MAS). Applications of NMR in biology. 1. Protein structure determination. Used to solve 3D structures of small to medium-sized proteins and LT, 30 kDa. Helps in understanding protein folding, stability, and conformational changes. 2. Drug discovery and binding studies identifies drug-protein interactions by monitoring chemical shift changes. Techniques like STD-NMR, saturation transfer difference NMR, help in screening potential drug candidates. 3. Metabolomics and biomarker discovery. Used to analyze metabolic profiles in biological samples like blood, urine, and tissues. Helps in detecting disease biomarkers, e.g., cancer, diabetes. 4. RNA and DNA structure analysis. NMR reveals base pairing, secondary structures, and interactions in nucleic acids. Useful in studying RNA AP tamers and ribo switches. 5. Enzyme mechanism studies. Tracks substrate binding, catalytic intermediates, and conformational changes in enzymes. Advantages of NMR spectroscopy: non-destructive, samples remain intact for further studies. Provides atomic resolution data, essential for structural biology. Works in physiological conditions. Unlike X-ray crystallography, NMR can study molecules in solution. Detects molecular interactions in real time, ideal for drug discovery and biomolecular dynamics. Limitations of NMR spectroscopy: requires high sample concentration, typically in the EM range. Expensive and time-consuming, requires high field magnets and specialized expertise. Limited to small to medium-sized proteins, larger complexes, and GT, 50 kDa are difficult to analyze. Previously asked CSIR Net question CSIR NET 2021. Which of the following nuclei is commonly used in biomolecular NMR spectroscopy? A squared C. B cubed C to the power of 6 D cubed squared P. Answer: B cubed C. CSIR NET 2019. What is the role of NOESY in NMR spectroscopy? A. Identifies hydrogen bonding patterns in proteins. B. Provides distance constraints for 3D structure determination. C. Detects spin-spin coupling in small molecules. D. Measures molecular weight of biomolecules. Answer: B. Provides distance constraints for 3D structure determination. CSIR NET 2017. Which type of NMR technique is used to study protein-ligand interactions in drug discovery? A. COSY. B. STD-NMR. C. MAS NMR. D. Department NMR. Answer: B. STD-NMR. NMR spectroscopy is a crucial tool in molecular biology, providing detailed insights into biomolecular structure, function, and interactions. You said, ESR. ChatGPT said. Electron spin resonance ESR spectroscopy. Electron spin resonance ESR, also known as electron paramagnetic resonance EPR, is a spectroscopic technique used to study unpaired electrons in biological and chemical systems. Unlike NMR, which detects nuclear spins, ESR detects electron spins and is useful in studying free radicals, transition metal complexes, and metalloproteins. Principle of ESR spectroscopy. Unpaired electrons possess intrinsic spin and behave like tiny magnets. In an external magnetic field, the unpaired electron can occupy either a lower energy parallel or higher energy antiparallel state. When exposed to microwave radiation, the electron absorbs energy and transitions between spin states. The absorption signal is detected and plotted as an ESR spectrum, revealing information about the electronic environment. The resonance condition is given by H nu equals G beta B, where H equals Planck's constant, nu equals microwave frequency, G equals G factor depends on the electron's chemical environment, beta equals Bohr magneton, B equals magnetic field strength. Types of ESR spectroscopy. 1. Continuous wave ESR CWESR measures steady-state absorption of microwave radiation. Used for free radicals and metal complexes in biological systems. 2. Pulsed ESR. Uses short microwave pulses to measure relaxation times in electron interactions. Provides higher resolution and structural information on protein dynamics and spin-labeled molecules. 3. Electron nuclear double resonance ENDOR. Combines ESR and NMR to study hyperfine interactions between unpaired electrons in nearby nuclei. 4. Double electron resonance DER measures long-range distances between two unpaired electrons, useful in protein structural studies. Applications of ESR in biology 1. Free radical detection. Identifies reactive oxygen species ROS and reactive nitrogen species RNS in biological systems. Useful in studying oxidative stress in diseases like cancer and neurodegeneration. 2. Metalloprotein analysis. ESR detects transition metal ions in proteins e.g., Fe3, Cu2, Mn2. Helps in understanding metal enzymes like cytochrome C oxidase and superoxide dismutase SOD. 3. Protein structure and folding. ESR with site-directed spin labeling SDSL provides structural insights into membrane proteins and conformational changes. 4. Drug-protein interactions. ESR studies how pharmaceutical compounds interact with metalloproteins and radical species. 5. Radiation biology. Measures DNA and protein damage caused by ionizing radiation and environmental pollutants. 6. Lipid membrane studies. ESR spin labeling tracks lipid mobility and membrane dynamics in biological membranes. Advantages of ESR spectroscopy. Highly sensitive to free radicals and transition metal complexes. Can study biological samples without extensive preparation. Provides structural and functional insights into proteins and enzymes. Limitations of ESR spectroscopy. Only detects paramagnetic species, substances with unpaired electrons. Requires low temperatures for high-resolution measurements. Less commonly used compared to NMR and X-ray crystallography for biomolecular studies. Previously asked CSIR-NET question CSIR-NET 2021. Which spectroscopy is most suitable for detecting free radicals in biological systems? A. NMR. B. ESR. C. UV-Vis. D. Mass spectrometry. Answer. B. ESR. CSIR-NET 2019. What is the principle behind ESR spectroscopy? A. Absorption of microwave radiation by unpaired electrons in a magnetic field. B. Absorption of radio waves by atomic nuclei. C. Absorption of visible light by conjugated molecules. D. Emission of X-rays from heavy metals. Answer. A. Absorption of microwave radiation by unpaired electrons in a magnetic field. CSIR-NET 2017. Which biomolecule can be studied using ESR spectroscopy? A. DNA double helix. B. Free radical intermediates in enzymatic reactions. C. Ribosomal RNA. D. Lipopolysaccharides. Answer. B. Free radical intermediates in enzymatic reactions. ESR spectroscopy is a crucial tool in biophysics, structural biology, and free radical research, offering unique insights into electron-rich biomolecules and paramagnetic species. You said. Molecular structure determination using X-ray. ChatGPT said. Molecular structure determination using X-ray crystallography. X-ray crystallography is a powerful technique used to determine the three-dimensional structure of biomolecules such as proteins, nucleic acids, and small organic compounds. This method provides atomic-level resolution, making it essential for drug discovery, structural biology, and material sciences. Principle of X-ray crystallography. When a crystal is exposed to a beam of X-rays, the atoms in the crystal cause the X-rays to scatter. The scattered X-rays interfere constructively and destructively, producing a diffraction pattern. Using Bragg's law, the diffraction data is analyzed to reconstruct the electron density map, which reveals the molecular structure. Bragg's law, n lambda equals 2d sin theta where n equals integer order of diffraction. Lambda equals X-ray wavelength. D equals spacing between atomic planes in the crystal. Theta equals angle of incidence. Steps in X-ray crystallography. 1. Crystal growth. The molecule of interest must be purified and crystallized. Crystallization methods include vapor diffusion, microbatch, and seeding techniques. 2. X-ray diffraction. The crystal is exposed to a monochromatic X-ray beam. The diffraction pattern is recorded using a detector. 3. Data processing. The diffraction data is analyzed mathematically using Fourier transform to generate an electron density map. 4. Model building and refinement. A molecular model is constructed to fit the electron density. The structure is refined to improve accuracy. Applications of X-ray crystallography. 1. Protein structure determination. Provides atomic resolution structures of proteins, enzymes, and receptors. Helps in understanding enzyme mechanisms, protein folding, and conformational changes. 2. Drug discovery. Determines how small molecules bind to proteins, aiding in rational drug design. Used to develop inhibitors for HIV protease, cancer targets, and bacterial enzymes. 3. DNA and RNA structure analysis. Led to the discovery of the double helix structure of DNA, Watson and Crick, 1953. Helps in studying RNA folding and ribosome structure. 4. Material science and nanotechnology used to study the structure of minerals, catalysts, and nanomaterials. Advantages of X-ray crystallography. Provides high-resolution atomic details. Works for large biomolecules that cannot be studied by NMR. No size limitation, unlike NMR spectroscopy. Limitations of X-ray crystallography. Requires high-quality crystals, which can be difficult to obtain. Cannot S. Molecular structure determination using X-ray crystallography. X-ray crystallography is a powerful technique used to determine the three-dimensional structure of biomolecules such as proteins, nucleic acids, and small organic compounds. This method provides atomic-level resolution, making it essential for drug discovery, structural biology, and material sciences. Principle of X-ray crystallography. When a crystal is exposed to a beam of X-rays, the atoms in the crystal cause the X-rays to scatter. The scattered X-rays interfere constructively and destructively, producing a diffraction pattern. Using Bragg's law, the diffraction data is analyzed to reconstruct the electron density map, which reveals the molecular structure. Bragg's law, n lambda equals 2d sin theta where n equals integer order of diffraction. Lambda equals X-ray wavelength. D equals spacing between atomic planes in the crystal. Theta equals angle of incidence. Steps in X-ray crystallography. 1. Crystal growth. The molecule of interest must be purified and crystallized. Crystallization methods include vapor diffusion, microbatch, and seeding techniques. 2. X-ray diffraction. The crystal is exposed to a monochromatic X-ray beam. The diffraction pattern is recorded using a detector. 3. Data processing. The diffraction data is analyzed mathematically using Fourier transform to generate an electron density map. 4. Model building and refinement. A molecular model is constructed to fit the electron density. The structure is refined to improve accuracy. Applications of X-ray crystallography. 1. Protein structure determination. Provides atomic resolution structures of proteins, enzymes, and receptors. Helps in understanding enzyme mechanisms, protein folding, and conformational changes. 2. Drug discovery. Determines how small molecules bind to proteins, aiding in rational drug design. Used to develop inhibitors for HIV protease, cancer targets, and bacterial enzymes. 3. DNA and RNA structure analysis. Led to the discovery of the double helix structure of DNA, Watson and Crick, 1953. Helps in studying RNA folding and ribosome structure. 4. Material science and nanotechnology used to study the structure of minerals, catalysts, and nanomaterials. Advantages of X-ray crystallography. Provides high-resolution atomic details. Works for large biomolecules that cannot be studied by NMR. No size limitation, unlike NMR spectroscopy. Limitations of X-ray crystallography. Requires high-quality crystals, which can be difficult to obtain. Cannot study dynamic molecular changes. Provides only a static snapshot. X-ray radiation can damage biological samples. Previously asked CSIR Net question CSIR NET 2022. Which of the following is the primary requirement for determining molecular structure using X-ray crystallography? A. Aqueous solution of protein. B. Crystalline form of the molecule. C. High salt concentration. D. Low pH environment. Answer. B. Crystalline form of the molecule. CSIR NET 2019. Which property of X-rays is utilized in X-ray crystallography? A. Ionization potential. B. Diffraction by crystal lattice. C. Fluorescence emission. D. Radioactive decay. Answer. B. Diffraction by crystal lattice. CSIR NET 2017. What is the purpose of Fourier transform in X-ray crystallography? A. To determine molecular weight. B. To reconstruct the electron density map. C. To measure fluorescence intensity. D. To identify protein sequence. Answer. B. To reconstruct the electron density map. X-ray crystallography remains a gold standard for molecular structure determination, playing a crucial role in biology, medicine, and material sciences. You said. Diffraction. ChatGPT said. Diffraction, principles and applications. Diffraction is the bending and spreading of waves when they encounter an obstacle or pass through an aperture. It occurs for all types of waves, including light, X-rays, electrons, and sound waves. In biological and molecular studies, diffraction is crucial in techniques like X-ray crystallography, electron diffraction, and neutron diffraction to determine atomic and molecular structures. Principle of diffraction. When a wavefront encounters an obstacle or slit comparable in size to its wavelength, it spreads out. The extent of diffraction depends on the size of the aperture relative to the wavelength of the wave. Constructive and destructive interference of diffracted waves produces a diffraction pattern. Bragg's law and X-ray diffraction. In X-ray crystallography, diffraction occurs when X-rays interact with a crystal lattice. The relationship between the diffraction angle and atomic planes is given by Bragg's law. N lambda equals 2 d sin theta. N lambda equals 2 d sin theta where n equals order of diffraction. Lambda equals wavelength of incident X-rays. D equals distance between crystal lattice planes. Theta equals angle of diffraction. This equation is fundamental in determining molecular structures in crystallography. Types of diffraction. 1. X-ray diffraction XRD. Used to determine atomic structures of proteins, DNA, and small molecules. Crystals produce a diffraction pattern, which is analyzed to reconstruct molecular structures. 2. Electron diffraction. Uses electrons instead of X-rays, which have a much shorter wavelength, providing high resolution. Used in studying thin films, nanomaterials, and viral structures. 3. Neutron diffraction. Uses neutron beams to analyze structures of materials containing light elements like hydrogen. Particularly useful in protein hydration studies and membrane proteins. 4. Optical diffraction. Occurs when light passes through slits, producing an interference pattern. Used in laser diffraction techniques for measuring particle sizes. Applications of diffraction in biology. 1. Protein structure determination. X-ray diffraction helps in solving high-resolution 3D structures of proteins. Example, determination of hemoglobin and insulin structures. 2. DNA structural studies. Rosalind Franklin's X-ray diffraction patterns led to the discovery of the double helix structure of DNA. 3. Virus structure analysis. Electron diffraction has been used to determine capsid structures of viruses like poliovirus and Zika virus. 4. Nanomaterial characterization. Diffraction techniques help study nanoparticles, carbon nanotubes, and semiconductors. Advantages of diffraction techniques provides high-resolution structural information. Non-destructive technique for analyzing biological macromolecules. Essential for drug discovery and material sciences. Limitations of diffraction techniques requires high-quality crystals in X-ray diffraction. Cannot capture dynamic molecular changes. Interpretation of diffraction data is complex and requires computational modeling. Previously asked CSIR Net question CSIR NET 2021. Which physical principle is used in X-ray crystallography for molecular structure determination? A. Absorption. B. Diffraction. C. Fluorescence. D. Conductance. Answer. B. Diffraction. CSIR NET 2019. Bragg's law is applied in which of the following techniques? A. UV vis spectroscopy. B. X-ray diffraction. C. Mass spectrometry. D. NMR spectroscopy. Answer. B. X-ray diffraction. CSIR NET 2017. Why is electron diffraction preferred over X-ray diffraction for studying thin films? A. Electrons have shorter wavelengths than X-rays. B. X-rays cannot penetrate thin films. C. Electrons have no charge and do not scatter. D. X-rays damage biological samples more than electrons. Answer. A. Electrons have shorter wavelengths than X-rays. Diffraction remains a fundamental principle in structural biology, allowing scientists to understand the atomic architecture of molecules. You said. NMR. ChatGPT said. Nuclear magnetic resonance NMR spectroscopy. Nuclear magnetic resonance NMR spectroscopy is a powerful technique used to study the structure, dynamics, and interactions of molecules at the atomic level. It is widely used in structural biology, organic chemistry, and drug discovery for characterizing proteins, nucleic acids, and small biomolecules. Principle of NMR. Atomic nuclei with an odd number of protons or neutrons, e.g., H cubed C to the power of 5n cubed p, possess a nuclear spin. When placed in a strong magnetic field B, these nuclei align either parallel, low energy, or anti-parallel, high energy, to the field. Applying a radio frequency RF pulse excites these nuclei, causing them to transition between energy states. As the nuclei return to equilibrium, they emit RF signals, which are detected and transformed into NMR spectra. Types of NMR spectroscopy: 1. Proton NMR, HNMR. Detects hydrogen nuclei, commonly used for organic compound analysis. Provides information on chemical environment, functional groups, and hydrogen bonding. 2. Carbon-13 NMR, cubed C NMR. Used to analyze carbon skeletons of organic molecules. Less sensitive than HNMR but provides detailed structural information. 3. Heteronuclear NMR to the power of 5n, cubed p, etc. Used to study nucleic acids, RNA, DNA, and phosphorus-containing compounds. 4. Two-dimensional 2D NMR. Techniques like COSY, NOESY, HSQC, and TOCSY help determine protein structures and molecular interactions. NOESY is particularly important for protein-ligand interactions. 5. Solid-state NMR. Used for studying membrane proteins, fibrils like amyloids, and insoluble biomolecules. Applications of NMR in biology: 1. Protein structure determination. NMR helps determine 3D structures of proteins in solution, unlike X-ray crystallography, which requires crystals. Used for dynamic studies of protein folding and conformational changes. 2. Drug discovery. Helps in studying protein-ligand binding for drug design. Used in fragment-based drug discovery, FBDD. 3. Metabolomics. Used to analyze metabolic pathways and disease biomarkers. Helps in understanding cancer metabolism and neurodegenerative diseases. 4. Nucleic acid studies. Provides insights into RNA folding and DNA-protein interactions. Advantages of NMR spectroscopy: non-destructive and requires no crystallization. Provides information on molecular dynamics. Can analyze molecules in solution, mimicking physiological conditions. Limitations of NMR spectroscopy: low sensitivity compared to mass spectrometry. Requires large sample quantities, especially for biomolecules. High-field NMR spectrometers are expensive and require cryogenic cooling. Previously asked CSIR-NET question: CSIR-NET 2022. Which of the following nuclei is commonly used in biomolecular NMR spectroscopy? A to the power of 4n. B cubed C to the power of 8 D squared C. Answer: B cubed C. CSIR-NET 2020. What is the function of NOESY in NMR spectroscopy? A. Determines molecular weight. B. Provides structural information based on nuclear Overhauser effect. NOE. C. Measures fluorescence emission. D. Identifies functional groups in infrared spectra. Answer: B. Provides structural information based on nuclear Overhauser effect. NOE. CSIR-NET 2018. Why is cubed C NMR less sensitive than HNMR? A. Cube C has a lower natural abundance. B. Cube C absorbs lower energy photons. C. Cube C does not interact with a magnetic field. D. Cube C has a larger atomic radius. Answer: A. Cube C has a lower natural abundance. NMR spectroscopy remains an essential tool in structural biology, chemistry, and medicine, helping researchers study biomolecules at atomic resolution. You said. Diffraction spectroscopy. ChatGPT said. Diffraction spectroscopy. Diffraction spectroscopy is a powerful technique used to analyze the atomic and molecular structure of materials based on the diffraction of electromagnetic waves, such as X-rays, or particles, such as electrons or neutrons. It is widely applied in structural biology, crystallography, materials science, and nanotechnology. Principle of diffraction spectroscopy. When waves, X-rays, electrons, or neutrons interact with a periodic structure, such as a crystal lattice, they scatter in various directions. The scattered waves interfere constructively or destructively, producing a diffraction pattern. The intensity and position of the diffracted beams reveal information about atomic arrangement, interatomic distances, and molecular structure. The key mathematical relationship governing diffraction is Bragg's law. N lambda equals 2d sin theta. N lambda equals 2d sin theta, where n equals order of diffraction. Lambda equals wavelength of incident waves. D equals interatomic plane spacing. Theta equals angle of incidence. By analyzing the diffraction pattern, researchers can reconstruct the atomic structure of a sample. Types of diffraction spectroscopy: 1. X-ray diffraction, XRD. Spectroscopy uses X-rays to analyze crystalline structures. Applied in protein crystallography, mineral analysis, and drug design. Example: determination of DNA double helix structure by Rosalind Franklin's X-ray diffraction patterns. 2. Electron diffraction, ED. Spectroscopy. Uses electrons as the diffraction source due to their shorter wavelength, which provides higher resolution. Used in studying thin films, nanomaterials, and viral structures. Techniques include transmission electron microscopy, DEMT diffraction, and low-energy electron diffraction, LEED. 3. Neutron diffraction spectroscopy. Uses neutrons, which interact with atomic nuclei instead of electron clouds. Useful for studying light elements like hydrogen, making it ideal for hydration studies in biomolecules. 4. Optical diffraction spectroscopy uses visible light diffraction to study microscopic structures. Applied in laser diffraction particle size analysis and biological imaging. Applications of diffraction spectroscopy: 1. Protein and nucleic acid structure determination. X-ray crystallography helps determine 3D structures of proteins, DNA, and RNA. Example: structure of hemoglobin and insulin determined using XRD. 2. Drug discovery. Used to design new drugs by studying the structure of target proteins. Example: X-ray diffraction studies on enzyme inhibitor complexes. 3. Nanomaterial characterization. Electron and neutron diffraction analyze nanoparticles, carbon nanotubes, and quantum dots. 4. Virus structure analysis. Helps determine capsid organization of viruses like Zika and poliovirus. 5. Forensic science and archaeology. XRD is used in mineral identification and authenticity testing of ancient artifacts. Advantages of diffraction spectroscopy: provides high-resolution structural data. Non-destructive technique. Can analyze both organic and inorganic materials. Essential in structural biology, materials science, and nanotechnology. Limitations of diffraction spectroscopy requires highly ordered crystalline samples, X-ray diffraction. Sample preparation, e.g., crystallization of proteins, is difficult. Radiation damage can occur in X-ray diffraction. Interpretation of diffraction patterns requires complex mathematical modeling. Previously asked CSIR net question CSIR NET 2022. Which type of diffraction spectroscopy is used to determine protein crystal structures? A. Optical diffraction. B. X-ray diffraction. C. Neutron diffraction. D. Electron diffraction. Answer: B. X-ray diffraction. CSIR NET 2020. Bragg's law is fundamental in which diffraction-based technique? A. UV-Vis spectroscopy. B. X-ray diffraction spectroscopy. C. Fluorescence spectroscopy. D. Raman spectroscopy. Answer: B. X-ray diffraction spectroscopy. CSIR NET 2018. Which diffraction technique is best suited for analyzing hydrogen-containing biomolecules? A. X-ray diffraction. B. Electron diffraction. C. Neutron diffraction. D. Raman spectroscopy. Answer: C. Neutron diffraction. Diffraction spectroscopy remains an essential tool in structural biology, drug discovery, and materials research, providing insights into atomic and molecular organization. You said. Molecular analysis using light scattering. ChatGPT said. Molecular analysis using light scattering. Light scattering techniques are widely used in biophysical and biochemical research to analyze molecular size, shape, interactions, and dynamics in solution. These methods rely on the interaction of light with particles or molecules, leading to the scattering of light in different directions. The analysis of scattered light provides valuable insights into molecular weight, size distribution, and aggregation states. Principle of light scattering. When light encounters a molecule or particle, it can be absorbed, transmitted, or scattered. The intensity and angle of scattered light depend on particle size, shape, and refractive index. Different scattering techniques analyze molecular characteristics based on elastic, no energy change, or inelastic energy change scattering. Types of light scattering techniques. 1. Dynamic light scattering, DLS, also known as photon correlation spectroscopy, PCS. Measures Brownian motion of particles in a solution, estimating hydrodynamic radius and size distribution. Commonly used for protein aggregation studies, nanoparticle characterization, and polymer analysis. 2. Static light scattering, SLS. Measures the intensity of scattered light at different angles to determine molecular weight and shape. Used in protein characterization and polymer science. 3. Rayleigh scattering. Occurs when particle size is much smaller than the wavelength of incident light. Explains the blue color of the sky and is used in small molecule analysis. 4. Raman scattering. Involves inelastic scattering, where light energy changes due to molecular vibrations. Used in chemical fingerprinting and molecular identification. 5. Mie scattering. Occurs when particle size is comparable to or larger than the wavelength of light. Applied in cell and tissue imaging. Applications of light scattering in molecular analysis. 1. Protein and nucleic acid characterization determines molecular weight, shape, and aggregation states of proteins and DNA. Helps in studying protein folding and misfolding diseases, e.g., Alzheimer's disease. 2. Nanoparticle and polymer analysis used to measure size distribution and stability in colloidal systems. Helps in designing drug delivery nanoparticles. 3. Drug-protein interactions. Used in pharmaceutical development to analyze drug binding and stability. 4. Environmental and atmospheric studies. Rayleigh and Mie scattering are used in aerosol and pollution analysis. Advantages of light scattering techniques: non-invasive and requires minimal sample preparation. Provides real-time analysis of molecules in solution. Can detect molecular interactions and aggregation states. Limitations of light scattering techniques: sensitive to sample impurities and polydispersity. Requires precise calibration and optical alignment. In biological samples, background noise can affect accuracy. Previously asked CSIR net question CSIR NET 2022. Which light scattering technique is used to determine the hydrodynamic radius of molecules in solution? A. Raman scattering. B. Dynamic light scattering, DLS. C. X-ray diffraction. D. Circular dichroism. Answer: B. Dynamic light scattering, DLS. CSIR NET 2020. Mie scattering is observed when A. Particle size is much smaller than the wavelength of light. B. Particle size is comparable to the wavelength of light. C. Light is absorbed completely by the sample. D. Light does not interact with the sample. Answer: B. Particle size is comparable to the wavelength of light. CSIR NET 2018. Which light scattering method is commonly used to study protein aggregation? A. Raman spectroscopy. B. X-ray diffraction. C. Dynamic light scattering, DLS. D. Electron microscopy. Answer: C. Dynamic light scattering, DLS. Light scattering remains a crucial tool in biophysics, nanotechnology, and pharmaceutical research, providing insights into molecular behavior in solution. You said. Different types of mass spectrometry. ChatGPT said. Different types of mass spectrometry. Mass spectrometry, MS, is a powerful analytical technique used to determine the mass-to-charge ratio, MC, of ions, providing insights into the molecular weight, composition, and structure of compounds. Different types of mass spectrometry techniques are used depending on the sample type, required resolution, and analytical goals. 1. Time of flight mass spectrometry, TOFMS. Measures the time taken by ions to reach the detector after acceleration in an electric field. Principle: lighter ions travel faster than heavier ones, allowing separation based on mass. Applications: used in proteomics, biomarker discovery, and environmental analysis. Advantages: high resolution, fast analysis, and broad mass range. 2. Quadrupole mass spectrometry, QMS. Uses 4 parallel rods with an oscillating electric field to filter ions of a specific mass-to-charge ratio. Principle: only ions of a selected MC pass through, while others are filtered out. Applications: drug testing, forensic analysis, and gas chromatography. Mass spectrometry, GCMS. Advantages: high sensitivity, selective ion monitoring, and compact design. 3. Ion trap mass spectrometry, ITMS. Traps ions using electromagnetic fields and selectively ejects them for detection. Principle: ions of different masses are sequentially ejected and detected. Applications: metabolomics, pharmaceutical analysis, and food safety testing. Advantages: high sensitivity, ability to perform MS per megasiemens, tandem mass spectrometry, and compact size. 4. Fourier transform ion cyclotron resonance mass spectrometry, for ICRMS, uses a strong magnetic field to trap ions and measure their cyclotron frequency. Principle: the frequency of ion motion is proportional to mc, allowing ultra-high resolution measurements. Applications: structural elucidation of biomolecules, proteomics, and complex mixture analysis. Advantages: extremely high resolution and accuracy, useful for studying large biomolecules. 5. Orbitrap mass spectrometry. Uses a spindle-shaped trap where ions orbit around a central electrode. Principle: ion oscillation frequency is measured, providing precise MZ determination. Applications: proteomics, lipidomics, and pharmaceutical research. Advantages: high resolution, fast scan speed, and no need for cryogenic cooling. 6. Magnetic sector mass spectrometry. Uses a magnetic field to separate ions based on their mass-to-charge ratio. Principle: heavier ions are deflected less than lighter ones in the magnetic field. Applications: isotope analysis, organic compound identification, and radiocarbon dating. Advantages: high precision, useful for isotope ratio mass spectrometry (IRMS). 7. Tandem mass spectrometry (MS) for megasiemens or MS squared. 2 or more mass spectrometers are used in sequence to analyze fragmented ions. Principle: the first MS selects the ion, and the second MS analyzes its fragments. Applications: protein sequencing, post-translational modifications, and metabolomics. Advantages: high specificity, useful for studying complex molecules. 8. Matrix-assisted laser desorption ionization mass spectrometry (MALDI-MS) uses a laser pulse to ionize biomolecules embedded in a matrix. Principle: the matrix absorbs laser energy and transfers it to the sample, causing ionization. Applications: proteomics, peptide mapping, and microbial identification. Advantages: soft ionization method, minimal fragmentation, good for large biomolecules. 9. Electrospray ionization mass spectrometry (ESIMS) uses high voltage to create charged droplets, which evaporate, leaving gas phase ions. Principle: the solvent evaporates, leading to the formation of multiply charged ions. Applications: protein analysis, small molecule identification, and pharmaceutical research. Advantages: suitable for large biomolecules and non-volatile compounds. Applications of mass spectrometry: proteomics, identifying and characterizing proteins. Metabolomics, analyzing metabolic pathways and disease biomarkers. Environmental analysis, detecting pollutants and contaminants. Pharmaceuticals, drug discovery, quality control, and bioavailability studies. Forensic science, identifying drugs, toxins, and explosives. Previously asked CSIR net question CSIR NET 2022. Which mass spectrometry technique uses a laser for ionization? A. ESIMS. B. MALDI-MS. C. TOFMSD featuring ICRMS. Answer: B. MALDI-MS. CSIR NET 2020. Which MS technique provides ultra-high resolution by measuring ion cyclotron frequency? A. Quadrupole MS. B. Orbitrap MS. C. Featuring ICRMS. D. TOFMS. Answer: C. Featuring ICRMS. CSIR NET 2018. Which ionization technique in mass spectrometry is best suited for large biomolecules? A. Electron impact ionization. B. ESI. C. MALDI. D. Both B and C. Answer: D. Both B and C. Mass spectrometry remains an essential tool in biotechnology, pharmaceutical, and environmental sciences, offering precise molecular identification and analysis. You said. Surface plasma resonance methods. ChatGPT said. Surface plasmon resonance (SPR) methods. Surface plasmon resonance (SPR) is a powerful label-free optical technique used to study molecular interactions in real time. It is widely applied in biophysics, biochemistry, and pharmaceutical research to analyze biomolecular binding kinetics, affinity, and specificity. Principle of surface plasmon resonance (SPR). SPR is based on the resonance of free electrons plasmons on a metal surface when light is reflected at a specific angle. This resonance is sensitive to changes in the refractive index near the sensor surface, allowing the detection of biomolecular interactions. A sensor chip coated with a thin metal layer, usually gold, is used. A light beam is directed at the sensor surface, generating surface plasmons. When a ligand is immobilized on the chip and an analyte binds to it, the refractive index changes. This shift in resonance angle is detected and recorded as an SPR response, measuring interaction strength in real time. Types of SPR techniques. 1. Conventional SPR. Measures changes in light reflectance at a fixed wavelength. Applications: protein-protein, protein-DNA, and small molecule interactions. Advantage: high sensitivity in real-time kinetic measurements. 2. SPR imaging (SPRI). Uses an array-based sensor chip to detect multiple interactions simultaneously. Applications: high throughput screening and drug discovery. Advantage: can analyze multiple binding events in parallel. 3. Localized SPR (LSPR). Uses metallic nanoparticles instead of a continuous metal film, enhancing sensitivity. Applications: biosensing at the nanoscale, detecting low-abundance molecules. Advantage: highly sensitive and adaptable to various analytes. 4. SPR coupled with mass spectrometry (SPRMS) combines SPR detection with mass spectrometry for detailed molecular analysis. Applications: identifying unknown binding partners in complex biological samples. Advantage: provides both binding kinetics and molecular identification. 5. Plasmon enhanced fluorescence spectroscopy (PEFS) uses SPR to enhance fluorescence signals, improving detection limits. Applications: detecting ultra-low concentrations of biomolecules. Advantage: increased sensitivity compared to traditional fluorescence methods. Applications of SPR: drug discovery, identifying drug-target interactions and optimizing binding affinities. Protein-protein interactions, characterizing receptor-ligand binding kinetics. Antibody-antigen interactions, used in immunological assays and vaccine development. DNA-protein binding studies, investigating transcription factor binding to DNA. Pathogen detection, identifying viruses, bacteria, and toxins in medical diagnostics. Previously asked CSIR net question CSIR NET 2021. Which surface plasmon resonance method is most suitable for detecting low-concentration biomolecules? A. Conventional SPR. B. Localized SPR. C. SPR imaging. D. SPRMS. Answer, B. Localized SPR. CSIRNET 2019. Which of the following techniques is used for real-time, label-free detection of biomolecular interactions? A. ELISA. B. Western blot. C. Surface plasmon resonance. D. PCR. Answer, C. Surface plasmon resonance. CSIRNET 2017. What is the key physical phenomenon behind surface plasmon resonance, SPR? A. Light absorption by molecules. B. Electron resonance on a metal surface. C. Emission of fluorescence by biomolecules. D. Diffraction of X-rays through proteins. Answer, B. Electron resonance on a metal surface. SPR methods are essential in biomolecular research and pharmaceutical sciences, offering high sensitivity and real-time monitoring of interactions.